well, I guess we gotta figure out what you're doing.
Yeah.
I had a tech who started mid-February 2020. Which, I mean, I think in around September she finally gave notice because it was like she didn't have no bioinformatics background at all. Was a tech, was just getting started, and literally just had nothing to do for 6 months.
Wow.
I started around then. I had my first postdoc, and he was in the lab for like 4 months. And it was his visa was expiring. He was like, "I'm not doing it here."
Yeah, so the postdoc who wrote this started paper, we finally got really good at working together, and then her husband, who moved here with her from the Netherlands, was like, he had been trying to find work, but because the pandemic hadn't, and so he'd been a stay-at-home dad for 3 years at this point. It was just like, I'm not doing this. And so she moved back to the Netherlands.
Here's the microphone.
I actually much prefer having a lab like this because
That was granted. 458.
and then I took notes to my wife for now life.
And I think Scott's was in a little OPD.
I got a pair recently. I use it for running like 8:30 in the morning. Getting what I
More than done.
Hello.
And then if you need to like talk, I also have the earplugs. My figures.
That's too loud. How about that? Yeah. I can talk like a normal person and you can hear me. All right. That's
research strategy. I didn't realize that we scheduled for seminars and then our grad medical science educational departments for next version to the same time. So then this might be a little sparse. And then all the people that are doing research here, so it took
5 shots. A couple more minutes.
People filing it. And there might be some people also viewing on the live stream. Just say hi. All right. Yeah, so we'll get started. I know we unfortunately got a little conflict with GMS's faculty meeting, so yeah, so we need to watch out for what they do sometimes. But I'm Nelson Lau, the director of the Genome Science Institute, and it's my pleasure to be hosting Dr. Michael Baym from Harvard Medical School, whom I've actually never met in person, but just got acquainted through what was once the Twitter social media platform that's now degraded into a disaster. I've also interacted with Michael Baym on this mid-career PI Slack channel that he's also quite active on. And then, he's also now moved over to Blue Sky, which is now the current realm for where scientists are congregating if they want to waste a lot of time procrastinating, not writing their grants on social media.
A lot of time.
So, he's quite an interesting personality. He makes a lot of really interesting observations that gets a lot of retweets or likes or whatnot. But mostly I've been interested in his work in bacterial genetics and genomics, and he's from a department of biomedical informatics. And I thought that these themes really resonated well with what the GSI is promoting here on this medical campus, which is to promote genetics and genomics research. And I thought Michael Baym is a great speaker that I don't usually interact with because he works in the bacterial realm, but he's also worked with transposons and antibiotic resistance and virus discovery. So I thought that might be of interest to our research community. He's been an assistant professor at Harvard since 2017 and was just recently promoted to associate professor. He did his PhD with Bonnie Berger at MIT, so I share that comment. I also did my PhD at MIT and then did his postdoc training at Harvard with Roy Kashoni, where I think one of his landmark studies is this giant megaplate selection experiment testing different bacteria and how they in real time can grow from two ends of this giant plate till they come in. There's these like really great YouTube videos you can check out. He's had a number of Young Investigator Awards like the Pew Biomedical Scholarship and the Sloan Research Foundation Fellowship. And his lab has been funded by the NIH through a MIRA grant, as well as an NSF grant. So he's one of the very few people I know at a medical school whose work has been funded from the NSF. So I'd like to try to get that secret on how to do that here. Because usually when I talk to NSF, they like ignore us at a medical campus. Anyway, I'm looking forward to your talk, Michael, and you have the floor.
Thank you. Thank you. I don't know that I have anything to offer on the NSF thing besides like do things that medical schools care about less than you think.
Okay.
So I think I titled this like plasmid ecology or something like this. I want to tell you about some of the stuff we've been doing recently. So I think of myself, despite all of this, as an evolutionary biologist by sort of avocation. And because of this, I study microbes. So sort of very much for the same reason Willie Sutton, Robin Banks. If you want to look at evolution, I think you should look at microbes because that's where the evolution is. So I like starting by just sort of explaining this. Usually, this is either explaining why microbes to evolutionary biologists or why evolution to microbiologists, but I think to genomicists, this is also interesting because this is where a lot of the genetic diversity of the world really comes from, this engine of it. And it helps to think about, well, anyone who's done a transformation or any kind of genetics work, you've probably grown up a flask of E. coli at some point. Right? You start with a single E. coli, you stick it in a jug of LB, and about 13 hours later, you've got a liter of saturation culture. Now, if you let that keep going at exponential and you ignore physics and pretend that doesn't exist, after about 27 more hours, how big do you think it would be? Anyone want to take a guess?
We're in the realm of astronomy, probably.
You're exactly right. It is about the size of the Earth at that point. And about 16 hours later, if you really want to get into astronomy, it gets to the Schwarzschild radius and collapses back on itself as a black hole. But obviously other things have gone wrong before that. But the point is really that this kind of rapid exponential growth means that any kind of rare advantageous mutation, any sort of rare thing that this population happens to come across that allows it to take off in a new niche or gives it an advantage can really rapidly fill that niche, which allows all sorts of interesting vet hedging strategies to suddenly make a lot of sense. And sort of on a population scale, it can really use evolution as learning because you can wipe out half a flask of bacteria and 20 minutes later it's like nothing happened. And of course, this affects our lives too, as all of us remember almost exactly 5 years ago, there was this tweet, since as you know, I like Twitter and will call it that to its death. I'm going to read this. On 31st of December 2019, WHO was informed of cases of a new pneumonia of unknown cause in Wuhan City. A total of 44 cases have been reported. 11 are severely ill, 33 are in stable condition. So 44 cases back in December of 2019. And we all know exactly how that went and is still going as of October. This is another example where some sort of a virus mutated, made it into a new niche where it could spread, and that exponential spread went from barely hearing about it to shutting down our society in a matter of weeks. So where I came into this, though, and how I sort of made this transition from mathematics into experiments was this question of antibiotic resistance, right? And there's this classic story of antibiotic resistance, which I'm not going to spend too much time at, but I am going to just give you a little bit that made me start worrying about it, and hopefully will eventually infect you and make you start to worry about this as well. The classic story is, right, 1928, penicillin was discovered, introduced in 44. By 1945, there was resistance, but there were sulfonamides around. But a few years after that, resistance was reported. Tetracyclines, also resistance. And basically the central story of antibiotics that you hear every single time is as soon as we introduce a new antibiotic, resistance rapidly evolves. But you look at this and you sort of wonder, okay, so before tetracyclines were discovered, how could we know how rare resistance actually was? Right? Like, how do we know that there wasn't really resistance before it was discovered? Anyway, I'm just going to plant that there and then not really go into it just to get you doubting these things. So, back to evolution. Antibiotic resistance, classic example of Darwinian evolution, right? Sensitive population of the rare cell. I'm mostly just showing this because of Joy's incredibly cute graphics. But really classic, right? Rare resistant cell, some antibiotic pressure wipes out everything but that resistant cell, and now, like the E. coli in the new flask, it's able to spread, fill up the niche, and now you have a bunch of antibiotic resistance. And this happens genetically by two mechanisms. The first one that I started studying initially was spontaneous mutation, and I'm only going to show you that video that Nelson alluded to. I'm not going to actually get into it this much in the talk because I want to tell you about the other mechanism in a bit. But basically, this is just this idea that some sort of error in DNA replication introduces a mutation that allows more resistance, and bit by bit you can increase resistance until you get serious amounts. This has been done really classically by just plating and plating on a whole bunch of petri dishes. So I tried to figure out, okay, how old is this, right? How long have people actually been doing this? And the oldest one I could find was this paper from 1941. So this is the Florian chain paper for which they were awarded the Nobel Prize, where they first showed the industrial production of penicillin. And in one of the appendices, because papers were written real different back then, and this has like 14 different appendices, each of which could be its own paper. One of them, they showed the first treatment of a patient. And actually in another one, they showed that by serially passaging on increasing amounts of penicillin, they could get staph resistant to it. Turns out this is even older than that. So this is 1916 by Akatsu Noguchi on using arsenamides on spirochetes and evolving resistance that way. So it really, really very over 100 years old. And my work in it, we built this gigantic petri dish. This is very slightly larger than actual size, but not by a ton. It's like this big. And the way we set this up was no antibiotic on the outside. I know it looks like a football field, but they're going the other way. They're starting here in the end zone, where they're going to grow with no antibiotic, and then at each barrier, they're facing an exponentially increasing amount of antibiotics. In this video, 1 second is 8 hours. So they get to the line, and by this point, there's a big enough population that some mutant is able to make it through, which then again grows in population until it hits the next line, at which point, now there's other mutations that get selected for, and so on. And it really reliably, at least in trimethoprim and ciprofloxacin, gives you ridiculously high amounts of resistance in just, in this case, it was 11 days. But looking at this, there were a couple things wrong. I mean, the first one is predicts, you think, okay, resistance can evolve that easily. Why do antibiotics work at all? And the second thing is that if you actually go into clinics, look at what's the resistance is really causing problems, it's not really spontaneous resistance that's the issue. You know, trimethoprim resistance, yes, they can change their dihydrofolate reductase enough that it gets away with it, that it is able to escape the drug. But really what you care about is that these plasmids come in and carry a completely different dihydrofolate reductase that just doesn't care at all. And it's suddenly like the drug doesn't exist. And so this got me thinking about horizontal gene transfer and plasmids. And I also think this is interesting evolutionarily because unlike, in some sense, it's one of the few phenomena that we know is really important that just did not even exist in Origin of Species, right? This is a completely different mode of evolution where bacteria can share genes across really vast evolutionary distances. And I think I often see this particularly in undergraduate texts, talked about as bacterial sex, and I think that's really misleading because it's not recombination of similar organisms. I mean, these are, it's, Bill Hanage likes to describe this as, it's like you are gardening and you brush up against a flower and now your children and their children have petals, right? It's not that they're recombining, it's that a whole functional genetic set can move from an organism that has not shared an environment with this other organism for 1 billion years, can move to this organism and be fully functional. And so we started looking closer at plasmids because I at least hadn't really thought much about their biology, right? Okay, they sometimes are multi-copy. You'd like to think of them a lot because they carry genes. They carry virulence genes, they carry antibiotic resistance, sometimes metabolic pathways. They're really useful as tools. And that's sort of where we stopped thinking about them. But they self-replicate. And this means that they also evolve. And I want to tell you today 2 stories from my lab recently, stemming from asking questions about how plasmids evolve. The first one starts with some antibiotic resistance questions, which is really, can we find viruses that target them? Can we find viruses that interact with plasmids? And then the second one is sort of an oddball project that we've been working on for a while that I'm really just excited to talk about how plasmids themselves evolve inside individual cells, how they compete with each other. And so let me tell you about the first quickly. So the first one came from this question of how could a virus preferentially target antibiotic resistance? Say we wanted to do phage therapy, or we wanted to find phages. Wouldn't it be nice if we could find a phage that preferentially killed antibiotic-resistant bacteria? This is usually pretty hard because often, like, a virus coming in from the outside can't really see which mutant of DNA gyrase is active here, right? The virus has no way of knowing. Except when we look at how antibiotic resistance moves, if antibiotic resistance is on a plasmid, and that plasmid is able to move, usually that plasmid encodes a conjugate of pilus, which is basically this nanotube that it builds and shoots itself through, that gets expressed on the outside. Maybe we could find viruses that can find that and use it to kill the cell. And it turns out from looking at the literature, again, this started with my own naivety, there are a lot of these. And they actually come up a lot in biotech, but very often it's been forgotten that they're plasmid dependent. So M13, which is used for phage display, MS2, Q beta, all of these actually target the F plasmid conjugate of pilus. And then there was this weird one, PRD1, that's sort of an oddball that targets the INCP plasmid. And all of them work by targeting, so this one, side note, this one's actually really cool. It's actually exactly the same size as the pilus. And it seems like it basically hooks onto the end, and when the pilus gets retracted, it just follows it into the cell. PRDs hook onto the base and then use that to inject. And generally speaking, viruses have this whole fun evolutionary game where, they get one shot, right? A virus has to hook onto its target, and it gets to eject its DNA from itself once. If it does it wrong, it's dead forever. So it really, but if it waits too long, then it doesn't, then it gets degraded or something. So there's this whole arms race, even just on the surface of viruses, with them trying to figure out what's a good, reliable way to see a host and inject into it, and not just shoot my DNA out to nowhere or inject my DNA into a cell that it's not going to infect. This is a micrograph that actually some community college students took, we work with. This is the MS2 phage. So we put this with a conjugate pilus, and you can't see it terribly well on this screen, but basically this thing in the center that looks like a Brussels sprout, that is actually the conjugate pilus, just adorned with these viruses stuck to it. And they're sort of interesting because they've actually been shown that they can select against horizontal gene transfer, right? If you have a host that's got this plasmid, well, in order to get around the pressure from the virus, it has to evolve resistance in some way. And the two easiest ways of doing that are either silencing the pilus or kicking out the plasmid. If it kicks out the plasmid, now it's antibiotic sensitive, that's great. If it silences the pilus or breaks the pilus, now it doesn't move. So at least resistance can't transfer to different species. So this was sort of attractive for that reason. When we look historically, there's a bunch of pages that have been reported for different pilus types. We found, I think, 42 of them, or 40. Only 14 still remain. So, unfortunately, like many things in biology, as trends, as trends shift, labs close down, people's freezers get discarded, and many of these viruses we can't find again. So we were starting to wonder, okay, but these things seem like they're potentially really evolutionarily interesting, right? How common are they? Are they really these oddballs? Are they everywhere? How diverse are they? How do they change the evolution of resistance? And for some of them, how do they get into so many different bacteria? Or how are they able to infect so many different types of bacteria when most phages can infect like one substrain of E. coli and that's it? And so we had to rediscover them. And I want to tell you about that because this was sort of a fun project by my second graduate student, Natalia, there. So Natalia was saying, okay, how do you discover phages? Well, you go get some sand, something dirty. Wastewater is like, you call up, now we just call up Deer Island and like head over there and they give us jugs of it and it's great. In the pandemic, we had to be a bit more creative because they were busy giving all their wastewater to all the COVID people. And you say, okay, let's take a bacterium with this plasmid, we'll mix the two, we'll sort of mix the wastewater on a lawn and look for holes in the lawn. This is like classic Felix Durrell, they're called bacteriophages because it looks like something is eating the bacteria. And then you get a bunch of these plaques, and each one of these is a different virus, and so you take that virus and you test them with and without the plasmid, and most of them are hitting some host factor, and every now and then one of them doesn't grow without the plasmid, and that's the plasmid-dependent phage. Natalia looked at this and thought, this seems like a pain in the ass. If these things are really, really rare, I don't want to have to isolate thousands and thousands of phages just to not even find one. I would like to get a PhD at some point. And so we thought, how can we be a bit more clever about this? Well, one thing that's weird about these phages, that's really unique about them, is that they target unusually many bacteria. They target a really broad host range. And so we thought, what if we could use this to our advantage? What if we could somehow come up with a way of using that these viruses can infect a bunch of different bacteria to find them? And so Natalia and my former postdoc Sean, who now has their own lab at the Wadsworth in New York, came up with this assay where let's take two different bacteria that share the same plasmid. Let's co-culture them and then screen. And by looking at them, well, if it's just killing one or the other, so it's attacking a host factor, then it should only plaque in one of the two. But if it makes a complete plaque, then it can kill both of them. And so it's probably targeting the thing they have in common. And to sweep a year of work under the rug in half a slide, as is my prerogative as a PI, Natalia figured out you could get Salmonella enterica and Pseudomonas putea to grow next to each other. Getting two organisms that could grow at roughly the same rate on the same media, side by side, without killing each other, and were both genetically tractable, was not trivial. But when we put red and blue floors into them, actually it looked exactly, this was a year after the diagram. And now we've sort of scaled up, and all of the magic, all the fluorescent photos you see here, they're really just like this Canon camera with a filter wheel for amateur astronomers, some LEDs that we soldered up, and a computer controlling it. And in the pandemic, Sean and Natalia went around to every farm and wastewater treatment plant they could find, looking for these, figuring we're going to have to look for a ton to find any of these things, since only 6 of them were known. And we actually found it almost in the first place we looked, in a compost pile. And then we got some from the Muddy River. Because it's from the Muddy River, we called this one Big Pappy. And so it is in the paper now as PRD Big Pappy. And actually, in our initial screen, we found 55 phages that target Big P plasmid. And at that point, we stopped because we're like, okay, this is not what we were expecting. What's going on? And when we looked, there are a few interesting things that I can't really explain terribly well, but I just want to tell you about, because the first one is we sequenced a bunch of them. And so before we did this, there were 6 known worldwide from different continents, mostly from the 70s and 80s, really genetically similar despite being isolated across an ocean at different times. You don't usually see that with viruses. What we found in Boston wastewater fully encompasses that diversity. Like, the ones from Africa are not an outgroup. Or is it the one, right? They're just part of it. The funny thing, because their genetic structure is so conserved, when you have one, you're really able to tell it is what it is. So we looked in NCBI and we found one, this BCE1 phage, which is the only one we haven't actually done experiments with. It's called that because it's listed as a Burkholderia phage. Because whoever was using it probably had a Burkholderia that expressed an P. pilus, and they said, oh, it's a Burkholderia phage. It is absolutely a PRD. Like, no question. It's 99% identical, including synthetic. As a side note, once you have this working, it's actually sufficiently straightforward to do experimentally that we could have undergrads do it. So the last few years, Tom Bernhardt and I have been teaching a community college course based on the C-phages program. So the first year we were part of what we were doing was discovering plasmid-dependent phages. And actually 3 of the phages from the students made it into the paper. Going back, so there's this interesting structure to them. So there's pretty high nucleotide diversity. Like some of these proteins have, 15% different. Across every single one, they are fully synthetic. There are no accessory genes. So every single one of these phages that we found aligns end to end with no structural changes, no gaps, no new genes, nothing deleted. There's just a bunch of point mutations. They have really, really broad host ranges. So we got the INCP plasmid we were working with into 13 different species of bacteria. It's like usually when you say a broad host range phage, maybe it's like a couple of different E. coli and one salmonella. This is in Salmonella, Citrobacter, Enterobacter, Klebsiella, Serratia, Pseudomonas, Shewanella, and what you're looking at is these plaques, these holes, they're infecting all of them. Because not mirabilis for these four. To the point where we thought, okay, let's do all versus all. We ended up doing it in liquid, and so you can really see, we thought, all right, now we can understand host range. We can get all versus all, we can cluster it, we can do GWAS, and we got nothing. The only thing we were able to really understand about host range came from one of these phages that it didn't plaque on E. coli and Salmonella, so we got lucky finding it. It didn't plaque on E. coli and Salmonella, and so we were able to basically engineer in chunks to figure out of ones that did work to see what recombined to work. And so some of this seems to be the holon, but it can't all be the holon. They're really common in wastewater. So Site A and Site B, we were not allowed to put in print that we got it from Deer Island because that's one of their weird rules. We're allowed to get, these are from wastewater in Boston. There's only one wastewater treatment plant in Boston, but that's what we're allowed to say. Which has two catchments.
The North Shore and the South Shore?
Yeah, there's the North Catchment and the South Catchment, exactly. This is kind of interesting because E. coli phages are actually used as a metric of wastewater contamination of freshwater sources. And in fact, we're looking at only like a factor of 3 lower INCP-dependent phages. In fact, when you really look at this, there's more of them than there are Salmonella and Pseudomonas phages in wastewater. So at least in wastewater, basically, anything that was infecting our Pseudomonas, 99 times out of 100, was INCP dependent. There's something. So we went looking with metaviromics and didn't find them. And I think just there's an interesting cautionary tale here because I think there's some mix of they're small and not that common, but I actually think it's also because assemblers on reads do particularly badly in this circumstance because within each sample, assemblers work by assembling a genome, by piecing together a genome graph and looking for paths. When you've got a ton of synteny, but a bunch of pointwise diversity, you end up getting an incredibly sort of degenerate genome graph for a single contig. And I think part of what's going on is that they just, the population structure is almost perfectly designed to break genome graph assemblers. Works for other pilus types. We found this phage called FT Midnight, which is the first tailed phage that's known to infect conjugate pili. And you can read about this more. I'm going to move on to the other plasmid stuff because that's fun. But there's a bunch of them, super broad host range, sufficiently abundant to affect plasmid evolution, but how? So now we're working on the coevolution. And I think part of what I appreciate about this is especially personally coming from more of a genomics background, we couldn't have found these by genomics. We couldn't have found these by metagenomics alone. They're common, they are evolutionarily important, and they're just not enough of the DNA out there that you really catch them in a net. So,
any other T phages like T4, T7?
oh, those we catch.
Those you catch?
Those you catch. And the last thing. So, my grad student Ellie realized you can actually use this as a phage discovery technique, a modification of this for anything. So, here is a plate in which she mixed synthetic knockouts of 3 different phage receptors, each with a fluorescent tag, and then put wastewater on it. And you can see for Tol-C, that's a Tol-C dependent phage because the Tol-C knockout grows. Tol-C is interesting because it's an antibiotic efflux pump. Fu-A and TSX, other ones, and so you can use this as a general way of finding interesting rare viruses that interact with systems of interest. And we just pre-printed that like 2 weeks ago, speaking of the flurry of end-of-year papers. I'm definitely guilty. All right, so looking a bit closer at plasmids. So plasmids are really common. So Celia looked at this data from the Army, where there's this wonderful paper in which they sequenced every E. coli from Walter Reed Hospital for a year. Just every single one that showed up in the Walter Reed microbiology lab, they sequenced it. Because they didn't really have that much of a scientific team, just a bunch of sequencing to do outbreak detection, they basically did some light epidemiology on it and just published it and put the data set out there. So Celia went in and looked for plasmids. Turns out, of just this one type of plasmid, 70% of bacteria carry it, of E. coli carry it. Many of them carry multiple, but it's even more complicated than that. This yellow here is all the same kind of plasmid. It just carries 3 plasmid origins. So it's technically in 3 incompatibility types at the same time. It's a mess. Let's just say they're everywhere. When we look historically, from the British Culture Collection, plasmid mobility, like antibiotic resistance seems to be sort of increasingly on plasmids over time, becoming more mobile. And so this is saying they're really common and they're changing. And so this got us to this question of how do new plasmids emerge and compete? So you have a multi-copy plasmid, and 2 things have to happen for a new variant to take over. The first one, what pops up inside a cell, first it needs to compete with these other plasmids inside that cell and take over. Then once it's done that, now this cell is sort of competing with the other cells around it to take over the population. This is most of what we look at right now. This is governed by things like virulence factors containing antibiotic genes, metabolic genes. But that isn't, right? This is not necessarily, unless it has some strongly dominant phenotype, it's not. It has to compete in here sort of more molecularly. And we thought, let's try to look at that. Let's see if we can measure that. There's a lot of beautiful theory on it because if you've ever done population genetics, one of the first things you do is like the Fisher-Wright model, right? You do all this beautiful modeling of populations of a small number of things in a very constrained environment. And like, this is so beautifully amenable to that theory that for 20 years there have been wonderful theoretical papers that all end with, wouldn't it be great if we could measure this? The problem is, you've got to, in order to measure what makes one plasmid more fit than another, you've got to be able to compete them against each other. And in order to do that in a reasonable way, you need to make a whole population of cells that has exactly the same ratio of the 2 plasmids, ideally 1-to-1, right up until you say, okay, now compete, at which point you have to release that constraint and let them all fight with each other and see who wins. You can imagine this is tough. So when Fernando joined my lab and proposed a way of doing this, I'll admit, I said, there's no way this is going to work, but I'll let you try for 6 months. Because what he proposed was, what if instead of introducing a balanced number of plasmids, we instead built a synthetic dimer of the two things you want to compete. Took out all the recombinases, so this is vaguely stable, and then magically came up with a way of inducing a recombinase exactly when we wanted it and never any other time. Again, you can see all of the ways that this isn't going to work. To his credit, after about 6 months, he actually got it to work. And among other things, one of the hardest things was actually finding a way to turn off recombinase, because we had it on an auxiliary plasmid, even on the chromosome, and the problem is you get some leaky expression. It's really, really hard to actually turn off a gene completely. And because there just aren't that many copies of this molecule, and if the recombinase even acts a couple of times, it splits and you're done. We really needed this to be totally, totally off. And Fernando took a look at the flip recombinase from yeast and realized, actually the one he's using is not the wild one. It's been stabilized. Oh, well, what has it been stabilized against? So we looked, and the wild one sort of worked at 37, but worse. And so then he went and looked into yeast and found, okay, but there's yeast that grow in cold temperatures. That's how lager beers are made. Let's find the lager yeast. That one has the same Cerevisiae flip. But if you look at its ancestor, which is from like the cold valleys of Patagonia, that hasn't seen metabolic temperatures in 50,000 years. So he literally just took that flip, recoded it for bacteria, stuck it in, and sure enough, it had no activity at 37 and works beautifully at 30. And so now we have this wonderful system where we can basically grow these up at 37, they'll stay dimerized, and then we pop them into the 30 degree, they'll split into monomers and compete. So, what do you expect to see? If you're used to dealing with eukaryotic chromosomes, you might expect something like this. That is not what happens. These are not F plasmids. They don't have a segregation system. Instead, you get a lot of randomness. And I wanted to tell a little bit at this point about how plasmid copy control works, because this ends up becoming a copy control story. So basically, every plasmid, and this is an abstraction of it, there are a couple different ways that this is done, but every plasmid is basically the same architecture. There is some origin of replication and some repressor. And the origin just sits there and says, I'm going to copy this plasmid. I'm going to copy myself. And the repressor says, okay, but let's look for enough copies of this repressor. And once there are enough copies of us, we're going to shut down everything. Right? And so as long as this system is the same, if you have two different plasmids, so this other one comes along with a different plasmid that has the same origin and repressor, well, these repressors see each other and say, okay, let's shut down everything. And so what happens is that the system is regulating not necessarily the plasmid copy number of the whole plasmid, but the number of repressors, essentially the number of copies of the repressor around. And this ends up being controlling the sum of the number of the two plasmids. So this is classical plasmid incompatibility, right? If another one comes along with a different repressor, it doesn't care at all. And so this is how plasmids have classically been categorized by incompatibility types. That because these two regulate the sum, well, say they regulate the sum of 6 copies. Eventually, just by random chance, there's going to be 6 of one and 0 of the other, at which point they never mix again. And so you sort of get this random demixing that happens. So going back this, right, you actually get this stochastic division, and then stochastic replication, and then stochastic division replication, and then stochastic division replication, and very, very quickly you get this demixing. You can also think of this as genetic drift inside individual cells, because it's the same exact thing as genetic drift, right? But we can also look at it. So here is a razor blade inoculation. This is, again, taken with that same camera. What we've done is we've taken dimers that are exactly identical, except one of them has M scarlet, the other has M watermelon. These are two fluorophores that are different colors but separated by 8 point mutations. You dip a razor blade, you touch a razor blade to the black agar, grow it for about 12 hours in 37, and then we put it in 30 degrees under the camera, and this is what happens. You get competition and eventually stochastic demixing. And it's cool because you can see, okay, out here we really get the cells with fixed plasmid types, and in this you get mixed. These are the mixes, and so there's some time when they're in the cell at the same time and potentially competing with each other. And after about 7 days, nearly everything is fixed. We also did the control. I love being able to show control data, and it's pretty, because here we said, okay, well, what is, do we really know that this yellow is actually yellow cells, or is it just red and blue cells on top of each other? So we just took 2 endpoints and we mixed them 50-50, and we ran this exact same thing, and sure enough, no, you really are actually seeing this demixing happening. Because we're also geneticists, we also measured it with DNA barcodes. So basically, as you monomer, so when you split the dimers, the recombinase, when it breaks open a recombinase site, it doesn't know which, it doesn't know that those 2 ends that it's putting back together came from the same molecule, right? They're further than 1,000 bases from each other, so thermodynamically they're essentially on different they're functionally on different molecules, and so it just grabs 2 things at random and sticks them together. Because these plasmids aren't that big, preferentially it'll put the same one back together, but every now and then it'll transiently redimerize. But it'll redimerize randomly. And you can use that to statistically sample inside individual cells to figure out what diversity is. Where basically, right here, it should redimerize about 50% of the time to be heterodimers and 50% homodimers. Here you expect homodimers much more, but heterodimers in proportion. And this is literally from basic population genetics, Hardy-Weinberg equilibrium, or as I prefer to call it, the binomial theorem. By just sequencing across these junctions and counting how many heterodimers and how many homodimers you get, you can measure in real time, statistically, how your population is demixing. And so what do you expect? Well, first, because you put the dimers in, at the beginning you expect everything to be dimers. Then you induce the recombinase, and it should very quickly drop to Hardy-Weinberg equilibrium, and then from there it should divide, and there's strong theory about this falling off exponentially because it's classic decay of heterozygosity. This is idealized. Real data, actually, this would look like that, which is really exciting. So we got, it says the recombinase really does work very quickly. It actually is exponential. We put it on a log-log plot. It's this beautiful line. And so then we started saying, well, it shouldn't just be a line. We should be able to predict the slope of that line. And we started modeling that. And there was a problem. Because every time we tried to model the slope of this line, we got something that looked like this. Every set of reasonable parameters segregated too fast. So what's going on? Well, the one thing that we found in the literature that could explain this was something called eclipsing, where there's a delay in replication. So in classic plasmid replication, which there's theory papers on about like the Polya-Ern model and all of this, basically when plasmids replicate, they're small. So extension of the polymer is very fast. It can happen in seconds. But initiation is what limits it. It's basically which one gets to start, how many times you get to start. So here, you've got a 50-50 shot of replicating the red or the blue first. Red replicates. Now, without any eclipsing, red is now twice as likely to replicate again. And so you expect whichever one goes first overwhelmingly wins. But if there's a mechanism that says, all right, once it replicates, let's take a pause. Now the blue has a chance to replicate. And then it gets to pause, and now the red replicates. And so if there's some mechanism like this that sort of makes the plasmid take a break before it replicates again, then you expect segregation to happen more slowly because there's some amount more balance that's enforced in what gets replicated. Turns out there is such a system. This is exactly the system that bacteria use to stop replication of their chromosome more than it should, the damn-methylase system, where basically there's a protein, there's a methylation site, and there's a protein that binds to the hemimethylated state. So you get a methylated protein, they're both methylated, it replicates, now each one has only one methylated strand, the protein grabs them, keeps them from replicating until it can methylate again. And so this delay allows for this and fits with theory. And the really fun thing, because we have a synthetic system, is we can take it out. And so, from here we can say, all right, let's take the hemimethylation sites out of the red plasmid. And you get that. Let's take the hemimethylation sites out of the blue. Now keep in mind, this is not changing the protein products at all. This is just messing with the methylation of the origin. So there's that. There's a system for suppressing competition. I have 10 more minutes. Yeah, I can show you this in 10 minutes. I'll show you in 5. We can talk for a few minutes. But we also thought, okay, but faster replicating plasmids still do have an advantage, particularly if we take out the methyl site, but even without it. And this is a weird thing we noticed right off the bat. Plasmids that are more transcriptionally active tend to lose. So this should set you should have alarm bells at this point because, right, the one that's expressing more red loses, the one that's expressing more blue loses. Natural thought, which is ours as well, is, well, that's because expressing a protein is a metabolic burden. Spoiler, it's not that. I mean, it is a metabolic burden, but it's not much of one. And the way that we got around this was to look at single cell dynamics. So it just so happened that one evening, someone who was working with this microfluidic device called the mother machine came by and said, hey, my rotation student's project just completely imploded. Do you have anything we can stick in the mother machine that glows? Do we ever? So the way it works is it's this microfluidic comb with basically these little wells that you spin bacteria into, and then you watch them grow where it's capped on one end and media flows by the other side. What this does is it means when the cells replicate up here, if one of the daughter cells grows faster, it doesn't matter because it just randomly picks one daughter cell, whichever one's higher up, that's the one that lives. And what this means is that the cellular growth rate gets completely removed from the equation. And so you can really look at what happens to the plasma. It turns out this is perfect for looking at what happens to the plasma. It's independent of what happens to the cell. So we stuck it in, and this is what it looked like at first. There's a bunch of different colors because you don't need that many wells to get statistics, but because it's microfluidics, there's a bunch of them. So we thought, let's see how many experiments we can get going at once. You run it a bit, and so each of these colors is one starting condition. We tried a couple different dimers, a couple different ratios, and then once you know what the starting condition is, then you can track it over time by inducing the dimer splitting, and then you can really watch single cell trajectories of the varying plasmid count. Yeah, it's a 1.6 million wells per experiment, which is, I think, somewhere in the range of like 1,000 times what we need. But they build this for other things, and we beg them for we just use it for like a day. And we find, sure enough, all right, these are two fluorophores on the same promoter. And it's an imaging artifact that these aren't exactly even. We go in and count it. It just doesn't see the M watermelon as well. But when we put the M scarlet on a weak promoter and the M watermelon on a strong promoter, now we actually get really, really strong bias. This was before we took out the hemimethylation sites. When you remove those, it's just like it crushes it. So there is a replication expression conflict in plasmids, at least in multi-copy plasmids. They seem to not like expressing resistance genes, or you can sort of think like small plasmids don't like expressing genes. And so the last thing we're doing now is saying, okay, well, what happens if we set up a situation where the cell wants one thing and the plasmid wants something else, by putting in a little bit of an antibiotic resistance gene in one of them versus the other. So now you have more expression, but it's expression of something the cell needs, and we can get all sorts of interesting things happening where as it grows, you get more of one than the other. Here in this case, it's hard to see here because this is old data before we took out the hemimethylation sites. But basically, at first in this drug concentration, the one that expresses the antibiotic resistance gene actually starts to lose, and then eventually it fixes randomly in a few cells and they take over and win. And you get all sorts of weird games in there. And I think part of, to me, it's a bit roundabout from here, but the reason, if you look at the plot past about there, this is saying the thing the cell wants always wins. If you take out the hemimethylation sites, you get a lot more plasmid level selection. And in fact, we get cases where the thing the cell wants doesn't win. And so part of what I think is happening is that this hemimethylation system is acting as almost domestication of plasmids, like turning them from parasites to symbiotes, by at least allowing a little bit more time for cell level selection to work. But we're still just playing with it, right? We just built this system that lets us look at something we've never seen. Now we're seeing what we see. So just to sum up, we found plasmids carry these mechanisms that partially mask the fitness differences within cell and shut down genetic drift a bit. The conflict biases which one wins. There are these conflicts that favors silent plasmids, but clearly that's not an ironclad rule because there are genes expressed on plasmids out in nature. Although there are a lot of I do wonder if this explains why there are these cryptic plasmids that are so common that seem to express nothing. That's just sort of an end state of the plasmid being this pure predator. And in weird cases, plasmids that confer a higher benefit to their host often have a harder time fixing in a population. Because like a plasmid that comes in and expresses a ton of a great gene that's more than the cell needs, once it's there at a small copy number, it's no longer dominant. Right, that phenotype's no longer dominant, and so additional copies just bear the cost but no additional benefit. And so you get this strange trade-off where you actually end up getting selection for sort of inefficient production of genes. I'm going to end there because I'm totally out of time, and also have no more slides. So I want to thank my lab. This is the lab as of last summer. Basically, everyone in the lab has data that came into this in some way, almost everyone. And of course, our various funders that, Nelson makes it sound very impressive, like we get funding from a bunch of places, whereas on a day-to-day basis, it feels more like we just sort of cobble together whatever works and hope for the best. So, be happy to take any more questions.
That was great. Any questions from the audience?
Yeah, that was very fascinating. My mind goes back to the fact that you found all of these phages that were originally described from the reaches of the global here in Bosnia, and there's the genetic similarities. But then you showed the pictures that the students had taken, the electron micrographs. They looked quite different.
Oh, those were just dirty electron micrographs.
Oh, okay.
Yeah, no.
So structurally, even though they have all
no, structurally, this is just junk attached to it.
I see.
Okay. No, these were. I mean, I am no electron microscopist. My first experience with electron microscopy was also in this course. No, when we get images that the core facility operators took, they totally identical. But good catch.
Okay, thanks.
So I guess one model interpretation of the promoter and replication is sort of that they're mutually exclusive. They can choose to transcribe or can choose to replicate. Does anybody have some sort of imaging system, give them some BRDU, give them some BRU, and see how much they overlap versus... Is that something they can do in any of these bacterial systems? I don't know.
I mean, this was literally figuring out what the nature of that replication of that conflict was literally the NSF grant that just got funded.
Okay.
Because Fernando and I have two different hypotheses on it that compete. His hypothesis that we're both coming around to the other ones in some way, but his original hypothesis was actually RNA polymerase and replication fork collisions causing fork stalling. We've come to think that's less likely just because of timing. Just because extension happens so fast that it really, most of the game should be initiation. And that one you can't do with those dyes quite so easily, right? Because what I think is actually happening is when the gene is transcribed, since it's a very small plasmid, you get a procession of supercoiling that even though it doesn't necessarily cause a damaging amount of supercoiling, it decreases binding of the replication promoter, or the replication primer, just a little bit. But that's enough to slow initiation and let something else go first. But yeah, we have a grant exactly on this of tracking the locations, trying to mess with supercoiling, and like building all these different permutations to figure out which one it is. I don't know yet.
Cool, thank you.
But yeah.
So this segregation of these recombined flipped plasmids seems really stochastic in your selection system that's 2D like this. If you did this in 3D culture, like liquid media, does it still partition so plainly, or do you?
This
go
is
like-
liquid.
Oh, this is all liquid. Oh, okay.
This is liquid. Yeah, all
is
this.
There, is there any difference in dynamics when you grow them 2D versus?
No, in
3D?
this case, there is some. So there is some difference from the geometry. Let me find it. You can actually see it on this. So there's a reason we did razor blades, which is that if you start with a single cell, a single colony, you end up with a circle. That makes things much harder to interpret. Because as you go vertically here, based on just how much is red and how much is blue, you can measure fitness differences. With the circle, it's much, much harder. And also with the circle in an expanding population, here you have here as you go up, you've got a relatively constant population size. With a circle, as you expand, the population gets bigger. And this means that, you know, drift doesn't work the way you think it does, because small things that sort of stochastically go to small numbers may still be expanding enough that they can survive. And so you don't get the same extinction dynamics because of that. So there is some geometry that matters here, but it's not 2D versus 3D. It's like, it's almost like 1D versus 2.
Okay. And then if you throw an insult like a bacteriophage on these systems, what happens? Do the plasmids still segregate out randomly, or does
So
the
these
phage?
particular plasmids are not mobile. We've stripped, I mean, these are like almost wholly synthetic plasmids. We've stripped everything out of them. They don't have their dimer resolution system. They don't have a pilus. They're not mobilizable. Some of what I want to start playing with is, yeah, if you start putting that stuff back in, what happens? So I don't expect them to interact with phage in any real way, because none of these dynamics are happening on the outside of the cell.
Okay.
Right? There's nothing, like the phage can't see color, right? So a phage hitting one of these cells would have absolutely no way of knowing whether it's red, blue, or yellow. And so you wouldn't expect a differential effect. I think it's a really interesting question. What happens when we start making these more and more like natural plasmids, right? We went with really, can we make the absolute minimum stripped down plasmid so that we don't get any weird biological things popping up that mess us up? And even then, we got this hemimethylation popping up that messed us up.
Yeah.
So,
Which was originally there to combat phage infection.
hemimethylation?
Yeah. No?
No, I'm not sure. I
Oh.
think
That's how I've always learned it. Yeah.
no. So this is not restriction modification related.
Oh, okay.
No, this is like it makes sure that you don't get runaway replication of the genome.
I see.
At least that's
Okay.
how I was taught.
Okay.
Damn methylase, but I should really read up on it.
I see.
No, this seems to be like an endogenous just genome maintenance methylation tagging.
Ah, okay. Mike?
Just have a quick one. Could you tell us more about like how the phages recognize the pilus sort of molecularly? Are they encoding like a pilus receptor? And
Yeah. That's. I mean, it's really, as far as I know, are any of these good enough? You can actually. None of these are quite good enough. You can see the spike proteins on them, but these PRDs, they really look a surprising amount like coronaviruses. That spike binds, at least for PRDs, to the base plate, and it's complementary to it. And somehow when it locks, it introduces a conformation change that acts as a trigger.
so you basically found all of the same type of receptor, right, in your phages. And do you think if you sample further afield, you find other classes?
I think based on the diversity that we found here, I think if we kept sampling with the same bait, we'd find the same thing. But one of the things we've been doing is trying to evolve plasmids that are resistant to phage infection and then go sampling with those. You know, it's the classic screen, you get what you select for, right? We got more of what we selected for than we expected. And I think if you went further afield, you'd probably find things that were further, but it's not clear how biologically significant it would be. And I think there's also, you know, there's interesting, where did my mouse go? Yeah, there's probably interesting questions of what are the different host ranges of these? How are they narrower versus broader with respect to each other? Is there this sort of classic trade-off between being a generalist who can infect all these different bacteria but isn't good at any one of them, versus being a hyper specialist? Yeah, again, like when we saw this, we had all these questions like that, and like we can get the genomes and just answer it, and then it just made no sense. So, and Sean is looking at some of that in her lab now. So,
Okay, thank you everyone. Thank you, Michael, for the big talk.